Hello, this is Mr. McFall. This is the overview for the Biomedical Innovations course for January 6th and 7th. So let's talk about what I'd like you to do prior to coming to class on Thursday. So we've started the Restriction Enzyme Challenge questions. So do not submit those until we've gone over some of them, and then we do Thursday evening. So just get what you can done. So try to finish as much as you can of 611 Restriction Enzyme Challenge questions. And we will have any questions that you want to go over. We'll go over those on Thursday. We will not start doing the full lab until Monday. So we're going to be prepared to do that. So this will be due Thursday. So then we're really going to be focusing on that lab going forward in each day that we're in class and doing steps in that lab activity. So 612 construction and cloning of recombinant DNA. Just a quick overview of the experiment. I'll show you a couple of slides from this. So the overview of the experiment that we're doing, the goal is to use recombinant. Think the word recombining DNA. So we have two different DNA pieces and we're recombining them to get a desired result. So we're going to do that by cloning the DNA fragment that we want. In this case, it's a kanamycin resistance gene. Talk about that in just a minute, but we're going to try to clone that fragment and then do the process of transformation of bacterial cells. Now, bacterial cells can become resistant through transformation, where they consume DNA from another source, through transduction, where something like a phage or a virus injects the DNA, or they also do that through the process of conjugation, where two bacterial cells trade information. You did a conjugation lab in medical interventions. And then we're going to map that recombinant plasmid. So we're going to take that plasmid out. So we're basically taking the plasmid, the vector from bacterial cells, the plasmid, little circular piece of DNA, and we're going to extract it, map it, and see if we were successful in doing that. Why do we care? Why is this important? Well, this is absolutely one of the most critical or central methods in biotechnology of producing a biological product is to use bacteria to produce that product. Things like insulin are produced this way. Lots of pharmaceuticals, lots of chemicals are produced this way. So module 1, we're going to be getting into module 1. So what I want you guys to be doing is focus on that module 1 organizer. But just before we get into that too much, I just want to show you, here's what that plasmid looks like. And you can see that picture also in the module organizer or the protocol that I gave you. But here's a bigger version of that. And so you can see all of these are different restriction enzyme sites. It's 3,000 base pairs. There's one restriction site for the enzyme EcoR1. It already has an ampicillin resistance gene and has already been cut with EcoR1 to open the circle. So it's actually a linear piece. So it's already been cut. So this piece of DNA is already cut. So this is what our vector is. That linear piece of DNA has already been cut with EcoR1. And then this is the gene that we're trying to insert into this plasmid. And so here you can see the gene length is about 1,300 base pairs long, and EcoR1 is on both ends. So we basically have everything cut the way we want it, so it's just a matter of putting those together. The polypeptide protein is coded for right here in this main protein coding sequence. In the middle, you have what's called promoter sequence. And so as DNA is doing transcription, as it's being transcribed, this is the start, because this is where the ribosome is going to bind. All right, and this is where the stop codon would be. So this is where that gene is. So you do want to have some, why is there some before and after? That's kind of like a buffer area. You always want to insert a little bit before and a little bit after in case there's part of that DNA that gets cut off. So that's where these restriction sites are. And it's also just happens to be where the sequence is located. So you don't want to pick a site that's right in the middle or something like that. Last year we did a paper plasmid lab. I don't know if you remember doing that. We had to pick a certain site that would be ideal for inserting it. And so what I would also like you to do in addition to finishing those problem sets, and we'll work on this in class a lot on Thursday, is I'd like you to read through. So we'll talk a lot about the problem sets on Thursday, and we'll do a lot of like tube labeling so that we're ready to go on Monday. So you'll kind of have your everything set up so we can move quickly on Monday. So we're kind of do a lot of pre-lab work on Thursday. But what I want in Canvas, I have put the digital copy of the lab background and protocol. I gave you the big paper packet of this, but this is the digital copy of those specific pages. So you can look at that and see it. It's a big document, but so you can go through and see those in color and see the details of that. So I think that would be really helpful. This is kind of an overview of the whole lab and what we're trying to do. Once again, we're trying to insert that kanamycin resistance gene into this particular plasmid and ligate it together with DNA ligase. So that's our goal. So the other thing that I would like you to do, basically before Friday, because Friday I'm going to give you more instructions, and we're maybe working on 2 different units at once, because normally we would just keep going through this, but with the days in between, there's not going to be lots for you to work on. So we're going to be doing some parallel activities that I'll start introducing Friday as well. But with this, you're going to read through starting where it says Module 1. So if you scroll down, this background information is important. I would like you to read through this, particularly starting here with creating recombinant DNA and understanding what's actually happening in this process. So scan through that, read through that, and try to understand what's going on. All this information is really going to help you as you do the reporting and everything like that for this. But as you go through, you see experiment overview. This talks about the objective to assemble and analyze DNA molecules in vitro, which means in the lab setting, using several recombinant DNA techniques, including gene cloning, plasmid extraction, and restriction enzyme analysis. And so here you see Module 1, ligation of the plasmid vector of the kanamycin resistant gene fragment. So if you go back to that module organizer, and I made that assignment, I updated that, and it is right here. So this module organizer, it's a Google Doc. So what I'd like you to do before, by the end of Wednesday, Thursday, is to have Module 1 described. This doesn't have to be incredibly long. Maybe some bullet points, 2 or 3 bullet points, or short paragraph of the goal of the module and what the expected results are from the module. So how do you do that? Pull up this version of the file. So you scroll to Module 1. Module 1 overview, which is on page 9 of your, it's not actually page 9, it's page 13. It's page 9 of the packet because I took some of the pages out. Page 13. This is an overview of Module 1. And really, this is the page you need to look at. These are the detailed protocols. And I'm going to ask you to look at those. That's going to be your homework for over the weekend, is understanding how to do the protocols for Module 1. And it's a multi-step process because we have to do some ligating, and then we also have to verify our results. So read through here, and this kind of tells you the overall background, kind of the goals for this. So summarize what the goal of the module would be, and then the expected results are. At the end of the module, what are we hoping to see? So what are we hoping to see in the gel? That may be a little more tricky to think about. So what are we trying to determine? Think back to your problem sets, the restriction enzyme problems, and that will probably help you with that. So at the end of this whole lab, this module organizer will be due. So just keep up with that, and you'll be fine. Don't go ahead yet. Let's kind of stay on the same point. So Module 1 will take us, into next week quite a bit. So like probably Monday and Wednesdays. That's why I said probably going to start, moving into another unit and doing another unit alongside this one. So we'll kind of be working in 2 different units at one time. So, but you can handle that, I'm sure. So if you have any questions, let me know. So just a reminder, as far as things to be working on, summarize this. By the end of Wednesday and Thursday, make sure you have your restriction enzyme challenge questions done. Make sure you've read the lab background in Module 1 and fill out the Module 1 part of the organizer. And remember, in class, we're going to be doing some pre-lab stuff and going over some of these restriction enzyme problems. So that is it. And let me know if you have any questions on Wednesday about any of this, and I'll see you on Thursday. Thank you.